This video is designed to talk about fermentation. Fermentation is the process that happens when your cells do not have oxygen available. Remember, when we're talking about the regular process of respiration, glycolysis is anaerobic, but the Krebs cycle and etc. are aerobic, meaning that they require oxygen. So if you're in a situation where oxygen is not present, your body has to switch over to another process because your cells cannot go a sustained period without ATP. Remember, we talked about what happens if you're undergoing a sustained period where you're not getting enough oxygen. This process can break down very, very quickly. And in order to sort of fend that off, fermentation takes place. There's two types of this. The first one is called alcoholic fermentation. So we have the formula for it here. This formula should look somewhat familiar to you because pyruvic acid is one of the products that we're seeing at the end of the process of glycolysis, and so is NADH. We'll get back to that in a minute with a separate picture, but then if we're looking at the other side, the product side, there's alcohol, carbon dioxide, and NAD+. Alcoholic fermentation takes place in things like yeast and bacteria, and we actually use this to do things like have bread rise. We also use this to make alcohol like beer and wine and spirits, things like that. So it's a process that's taken advantage of in making all kinds of different food products for us. We'll actually do a lab in class where we'll watch yeast undergo the process of fermentation, and we'll actually track it. The thing that we'll be tracking is this, carbon dioxide, how much CO2 they're producing, and how long it takes it to inflate a balloon to a certain diameter. So we'll spend some more time on that one when we get to the lab. The main one for us to talk about today is the idea of lactic acid fermentation. This is what's happening inside of your muscle cells when you're undergoing exercise that's anaerobic, when your body cannot keep up with the oxygen demand that you're placing on it. One thing I want you to notice about these formulas is that the reactant side, the left-hand side for both of them, is the same, pyruvic acid and NADH. What varies between the two fermentation formulas is the product side, what you're getting on the other end. Lactic acid is the main product that we're seeing in lactic acid fermentation. That's what makes like your muscles burn, and it makes things uncomfortable if you're exercising really, really hard. So like let's say you're sprinting for a long period of time, or if you're doing some kind of sustained strenuous activity where you feel your muscles burning, it's actually that lactic acid that's building up in there. The other thing is the NAD+. If we take a look at this other diagram, it'll make more sense why NAD+ is an important product for us. So this is coming straight out of your textbook. What I'm hoping when you first look at this is that you recognize that the left-hand side should be very, very familiar. Taking glucose, breaking it down using 2 ATP to yield pyruvic acid and NADH should sound pretty darn familiar to you. This is what's going on during the process of glycolysis. What we'll see here with fermentation is, remember, glycolysis works without the presence of oxygen. It works anaerobically, which means when the Krebs cycle and etc. break down, glycolysis keeps working. It just works a little bit differently. And the way it works is what produces the process of fermentation for us. So if we look through the diagram, we're starting off with the glucose molecule, just like we were before. You can see it sort of condenses this down into one step. We talked about getting 2 net ATP out of this process, so this is just representing that. You know, we pay in the 2 ATP to break the molecule, you get the 4 back, so to make things simple, your book is just representing this as getting 2 ATP back immediately from breaking down glucose. You also use up the NAD+ to make NADH. The problem is, think about where NADH is going. NADH is an electron carrier, so that's going to the electron transport chain. But if your cells are undergoing the process of fermentation, that means that the Krebs cycle and etc. are not working. This is where the right-hand side of the formula becomes important. Instead of sending pyruvic acid to the Krebs cycle, which would be senseless because the Krebs cycle isn't functioning now without oxygen, your body ends up breaking that down. It uses 2 NADH, it actually gives up the electron, remember because NADH is an electron carrier, to reform pyruvic acid into lactic acid. What that does is it reproduces an NAD+ that goes back to the beginning of this cycle. Now, if you're looking at this and sort of scratching your head, I understand. This entire process, we've been taking NAD+, turning it into NADH, and then sending that NADH to electron transport chain to make more ATP. Here it kind of seems like the opposite of that. The reason it's important for the cell to make NAD+ here is the cell actually starts to run out of NAD+ during the process of fermentation. And think about it for a second. Usually NAD+ is used to make NADH, which then goes to electron transport chain where it's used up again, and then the NAD+ comes back to the beginning to be recharged again. In this process, there is nowhere for the NADH to go, since electron transport chain isn't working. What that means is the cell very quickly uses up all of its NAD+. At that point, it can't break down glucose anymore. It can't get you these 2 ATP. Now you're really in trouble because you can't make ATP through the Krebs cycle. You can't make it through electron transport chain because you have no oxygen. If you run out of NAD+, now you're not going to be able to make any here, and at that point your cells can begin to die. So this is a very serious problem. The way that the cell gets around this is by using that NADH, again, to give up its electron to rearrange pyruvic acid into lactic acid, which makes your muscles burn, which is kind of unfortunate, but it's better than the alternative, right? It's better than not being able to produce ATP and to have those cells begin to die. So this process is interesting in that it's sort of like the solution that your cells have to when you're in that anaerobic environment. And I understand it runs a little counterintuitively to some of the other things that we've talked about this chapter. We'll go over this one a little bit more in class as well, but if you just think of it as a modified version of glycolysis that works when there's no oxygen, I think that's sort of an easy way of thinking about it. The last thing we're going to talk about is a comparison between each of these processes. We'll look at alcoholic fermentation, lactic acid fermentation, and then things that happen in both. The first thing to talk about under alcoholic fermentation is sort of obvious. We have to discuss the products. There are two main products on this side of the process. We get alcohol and CO2. There's a third product technically, but that one's going to end up going in the middle, so we'll put that under the both column. They both produce NAD+. Because remember that NAD+ is going to go back and get recharged again into an NADH in the beginning of the process. But just to finish off the alcoholic side, this happens in things like yeast and bacteria. On the lactic acid side, the far side, this happens in humans and other mammals. So it's certainly not something that's limited to us. And then the product over here that's unique is lactic acid. So honestly, with these, I mean, it's one of those great science terms where the name kind of gives away the important thing that you need to know. There's some other things that we talk about under the both column. Remember, the NAD+ that we discussed, this is a product. But there are some reactants that you should know. Remember, the reactants are the same for both. The reactants are going to be pyruvic acid, which I'll just call pyruvic in there, yet that's a U, and then NADH. The last thing to talk about then is the fact that these guys are both anaerobic processes. So they're both going on only when the cell does not have oxygen. Remember that one because these are sort of exceptions. These are both, again, modified versions of what's going on during the process of glycolysis. So hopefully this was helpful at the end here to sort of breaking down the differences between these processes. The main one that you really need to know, the main one that we're going to focus on, is the idea of lactic acid fermentation. If you understand that diagram on the other page where it was producing NADH that's being used up then to make more NAD+, that's really the key to understanding this one. So we'll spend some more time focusing on those things in class, but this is just the main overview. This will certainly keep you very well prepared. If you know the things on this list, we can have a more detailed conversation in class tomorrow. So make sure you answer the questions once the video is over, and I'll see you guys in class.